Okay, so we are live now. We will wait for a few minutes until people log in. So let's wait for a few minutes. We'll start the live stream now.
And do you want people to use the Q&A function?
Yes, so hello to everybody who are joining us. Yeah, so whatever makes you feel more interactive and be more productive for the presentation, yeah, feel free to request that or suggest it. It's totally fine. I've had talks where they actually use more the Q&A. I've had talks that they actually raise their hand and go live asking the question. So it's totally down to the presenter. Okay, my live is not responding. Trying to regulate the Facebook live sometimes is not. Okay. Okay, it's going live. Okay. So we can wait a few minutes more. It's live on Facebook now, so people can see us through Facebook. Hello, everybody.
Hello.
Yeah. Okay. Share. I'm gonna share it to the other groups, Cyprus Biological Society. So how many classes do you teach now?
So I teach the same class in the fall and in the spring. And then I do a couple of sections of it. So in the fall, I teach Introduction to Cell and Molecular Biology. And then in the, wait, no, that's the fall. And then the spring, I teach Introduction to Genetics. And because I'm in one of the residential programs, I have the same group of students all year, which is really nice. And my biggest class size is 20. So
well, that's a good amount of students.
Yeah, they keep me on my toes, but it's a lot of fun.
And Colorado must be wonderful throughout the year. So you have a mental break in between.
Yeah, Colorado is beautiful and it gets as much sun as everybody says it does. So I'm pretty happy. I like that the weather changes so much here. There's it's so dry that it gets big temperature shifts. So it can be really nice and hot during the day and then it'll get very cold at night. So it's nice to sleep and you'll have snow one day and it'll be 80 degrees the next. And
that's a huge digression, but I guess you can adjust at some point.
It just keeps it interesting. I like it.
I'm sure. So do you want to wait a few more? We have a few people that have logged in already. Or maybe we can slowly start and people, I see people joining us on Facebook too. So, okay, let's start. So hello to everybody that have joined us today. It's a pleasure to welcome you to another e-lecture by the Cyprus Biological Society in the series Excellence in Biology. So today's guest is Dr. Melanie Peffer, who will be talking about biology education, which is a vital point to teaching biology and how we understand biology. So Dr. Peffer has a bachelor's and a PhD in molecular biology from the University of Pittsburgh, and she completed a postdoc training in learning sciences at the Georgia State University. She's a teaching assistant professor at the University of Colorado Boulder, where she teaches introductory biology as part of the Health Professions Residential Academic Program. She's also a research scientist level 2 with the CU's Institute of Cognitive Science. Her research is focused on how people learn biology with an emphasis on teaching about the nature of science knowledge and how we can use educational technologies to assess students' understanding of science. Dr. Peffer is the author of the best-selling book, Biology Everywhere, How the Science of Life Matters to Everyday Life. Biology Everywhere is a journey through the science of life as told through our daily experiences. She was the 2021 High Plains Library District Foundation's Writer in Residence and spent her residency writing her first children's book, On the River. Dr. Peffer is an avid science communicator, as you will see now, a TEDx speaker, and has developed biology content for TED Education and authored 2 Coursera specializations. And I must say, I had the pleasure of working with Melanie back in our PhD days. So it's, I'm really happy to see her grow and contribute so much in both in the science and education field and in the biology that she loves so much and spreads the word about. So without further ado, I give you Dr. Peffer. The floor is yours.
Wonderful. Thank you, Maria, for that kind introduction. It's so wonderful to be here and catch up. And thank you, everybody, for signing on today. I know it's the end of the day for many of you, so I appreciate you joining us for a couple of minutes. So my talk today is roughly in 3 parts. I'm going to pause at the end of each part, and we'll have a couple minutes for questions there. And it's going to be somewhat autobiographical at the same time, because if you've been to any of my career talks, and you can look up some of my career talks online too, I've kind of gone around a little bit, and I've found that that's kind of normal. And it's interesting to see how everything that I've done over the last 10 years has kind of combined to where I'm sitting now, with my main focus being on science communication. Okay, so without further ado, I want to open my talk today with a story about Alex. So this is Alex. Alex may be familiar to you from students you've had, or mentees, or people you've run into in the general public, because Alex doesn't really like science. And part of the reason Alex doesn't like science is because they think it's just a bunch of facts that they have to memorize. And Alex decided not to get a COVID vaccine because their uncle says they're not safe. And Alex doesn't think we can really trust the scientific evidence because everybody who peer reviews is just bought off by some kind of company. And Alex has been really frustrated during the pandemic because the mask recommendations are changing. Why can't the World Health Organization or the Centers for Disease Control just follow the scientific method and give us an answer? And so maybe Alex feels familiar to you because Alex is based on real people. And so the first part of my talk is going to be on epistemological beliefs about science. And so that's beliefs about the nature of science knowledge and how that belief about the nature of science knowledge plays into science literacy. And so these blue bubbles all represent different epistemological beliefs about science. In the second part, I'll be introducing the Biology Everywhere project and people's emotions and attitudes towards biology and science. And then finally, in part 3, I'll be talking about my current work on how we can engineer science communication. And that brings all of these pieces together, from the research I've been working on since my postdoc to my work over the last 2 years on Biology Everywhere, and to where I finally landed now with science communication work. So how did I get interested in working with people like Alex, who have these kind of negative associations and beliefs about science? And so, as Maria mentioned, we were graduate students together at the University of Pittsburgh, and we were both working with the glucocorticoid receptor and neural stem cells. And I got about halfway through graduate school and realized that I was not doing what I wanted to be doing. I had these passions for science and research and teaching and education, and didn't know how to bring them together, which led me to start volunteering at the Pittsburgh Zoo. And that's where I developed the Sci simulations, which I'll talk more about in a minute. And that led to a postdoc in the learning sciences, and then eventually into a faculty position in the learning sciences. So this was one of my first conferences. I think it was my first conference as a faculty member. And it was in Belgium, and it was a really great experience. But that's me asking a question at my first big conference as a faculty member. So one of the projects I've worked on the last couple of years, and I'm not going to talk about it beyond this slide because I think it's too tangential to the talk, but I'm happy to answer questions about it later, is about my experience going from molecular biology into the learning sciences and how very different that was and how important it is. So I've got a couple of papers on it. I had an NSF grant on this as well. Happy to talk about it later if you're curious. Okay, so with that, let's talk a little bit more about epistemological beliefs about science. So there's 4 dimensions of epistemological beliefs about science. And why do we care about them? It's because our beliefs underlie what we do and how we learn science. And there's a lot of big science issues going on in society today. So we're all aware of the coronavirus in the US, and I know in the UK as well, about plastic bag bans and if those are actually greener. We'll talk more about those in a minute. Vaccines is something people talk about. Reusable products. This is a reusable diaper. The effects of climate change. And I include the wolves as well, because in Colorado 2 years ago was actually a ballot measure that everybody voted on if we were going to reintroduce gray wolves into Colorado. And it was very contentious. And so we run into these issues very regularly as part of our daily lives. And so how do people reason about these issues? And that's something I'll also come back to at the end of the talk with motivated reasoning. But for now, let's focus on epistemological beliefs. So I mentioned there's 4 dimensions to these. The first is source. That's where we get our information. We see a lot of bias come up with source. So in this case, Alex is more likely to believe her uncle because her uncle is seen as a trusted source as part of her in-group. So we're more likely to believe information from people that look like us, talk like us, belong to a similar political group, similar religious group. And so these are beliefs about what is a good source for science knowledge. Someone with a science background is more likely going to say, oh, I learned that at a seminar, or I read that in a peer-reviewed journal article, rather than some random family member told me this, and that's why I am doing this. We also have beliefs about how we justify science knowledge. And so this also fits into what counts as scientific evidence. So this idea that we can justify things based on our opinions versus evidence. And what is scientific evidence? What is good scientific evidence? There's also beliefs that people have about the certainty of scientific knowledge. And this is a big one, because how science is often presented in classrooms is that when we know something, we know it forever. And that knowledge doesn't change. And there's been some interesting stuff written up about textbooks in this, because when you see a big textbook, I always think of like the molecular biology of the cell, which weighs about 7 pounds. It seems like we know everything there is to know about cell biology, but at the end of the day, we don't. And so part of the power of science is that science knowledge changes in light of new evidence. And when people see science presented as just memorizing a bunch of facts, which gets into our last part of epistemological beliefs, then it's hard to see that science knowledge can and does change. And so finally, this fits into what's called the structure of scientific knowledge. And this also fits into biases as well, because we like things to be cut and dry, and that it's, this and then this and then this, and that is what's going on. And that's also often presented to people in terms of, oh, it's just a bunch of facts that I have to memorize, when actually information and how the world works is very interrelated. And so it's much more complicated than just a listing of facts or simple cause and effect relationships. So one of the problems with beliefs is, how do we measure beliefs? Because one of the biggest issues with assessing epistemological beliefs is that people often tell you what they think you want to hear rather than what they actually believe. And there's also the issue too that people's beliefs aren't always fully conscious. They'll act in a certain way without being aware of how these beliefs are playing out. And so I mentioned before about my SCI simulation. So SCI stands for Science Classroom Inquiry. It's a way of giving students an authentic inquiry experience in the confines of a typical classroom. And this was an idea that I had and developed at the Pittsburgh Zoo. So this is a picture from many years ago at the zoo with students actually doing the size simulation. So this came about because I was teaching at the zoo on the weekends and working on my dissertation work during the week and trying to figure out a way to bring what I was doing during the week in the lab and make it into a classroom experience. So this is a way of simulating that for students. It scaffolds authentic inquiry. So a lot of what students see in classrooms is what's called simple inquiry. So these like recipe-like labs where if you do this and then you do this, you get this result, which isn't how it works in the real world. They get to look at real world data, real world problems. And the important part of this is that they're real world problems that don't have solutions. There are things that scientists are grappling with or things that are controversial that we don't have an answer to. Because that again fits into that idea of the certainty of scientific knowledge, that we don't always have cut and dry answers. And even if we have cut and dry answers, they're always subject to revision in light of new evidence. So it gives this authentic nonlinear format. So students run into dead ends, just like we do in the real world, and have to go back and start over. And here's the kicker. Because they're doing it on a web app, on a computer application, we can capture data. And we can use these data traces to assess their inquiry and consequently their epistemology in real time. So rather than asking them what their beliefs are on a survey, we look at what they do in an authentic experience as a proxy for their beliefs. So I always am asked, what does a size simulation look like? So if you go to sci.colorado.edu, you can do one on your own. But in the meantime, we're just going to watch a quick video. This is the older build of the simulation. We've since had a rebuild of it, but it should give you an idea of what to look for. So here's the testing and hypothesis revision phase. So here's our test screen. Students are always asked, why do you want to do this test? They get some information. And then so we see that this participant then Googles some additional information, which we found to be a very key behavior. So now they're looking up what this virus is. And then again, looking up more information about morbillivirus. And so now they're saying their hypothesis is refuted. So they're going to generate a new hypothesis. Saying how they want to test it, why they want to do it. So we're back at the testing screen, like for more information. And now they're going to look at manatees, which is another marine mammal. So now looking up more information again. And now they're concluding that their hypothesis is supported. I'm going to do another test. And again, looking for information. So that website in particular is a citizen science website where people are reporting their findings. And then now they're ready to make their conclusions. So you can see how there's choice in what they look at and that they're also looking at real world data. They can generate a new hypothesis until they're finally ready to make their conclusions. Okay, so what do these data traces and learning analytics tell us about what people believe about the nature of science knowledge? And so I'm not going to go into depth into any of these papers. You're welcome to look them up. I'm happy to answer questions. But one of our big findings that ended up being a nominee for best paper was that there was very different language used by participants. So we looked specifically at the concluding phases and found that experts are more likely to use what linguists call hedges. And so that's where it's like support, may, could. Whereas novices don't use that language at all. They arguably use more definitive language like cause. And so this could be telling us about people's beliefs about the tentative nature of science knowledge, the certainty of it, that it can change over time because experts are more likely to use those hedges. We also saw very significant differences in investigative approach. So the use of that information that we saw in the video, how many tests they did, which tests that they did they do those tests in some kind of organized manner, or was it kind of just happenstance? And so this first paper from 2019 was looking at a very qualitative approach and seeing the practices of experts and novices. And so experts in this case are people who've published in the biological literature. And so we needed a way to do more high throughput analysis. And that's when we started moving into learning analytics, or using these data traces and machine learning to be able to assess these investigative approaches in a faster manner. Okay, any questions on this first part of my talk? And you're welcome to put them either in the chat window or in the Q&A box. And if you think of things as we go, you can always leave them in the Q&A box, and I'll have another pause for questions in a few minutes, and I can look at them then too.
So Melanie, I have a quick question. So for these simulations, from what age can kids participate?
Yeah, that's a great question. So I've done it with secondary school. So middle schoolers is the lowest I've gone. This data that I just showed was with undergraduates. And so I decided to, although I've tested it with middle school and high school, I decided to mainly focus on undergraduates because I didn't want content knowledge to be a factor in how people were doing the simulation. So I was looking for students who had more of a basic biology knowledge. Okay. And I also found, especially with the middle schoolers, they were much more likely to just do every single test because I think they felt nervous or uncomfortable about, they didn't want to miss anything. And that's telling us something completely different that doesn't have to do with their beliefs.
And did the kids have like a preferred subject, or were you driving the content per se?
I've tested a couple of different simulations. They've largely been a little bit more ecology focused. I had one on neural tube defects as a very early simulation, and we've got a current one right now that's a molecular biology simulation that I'm really excited about. I have a wonderful undergraduate mentee working with me right now, and she wrote a molecular biology scenario about the next pandemic.
Oh my!
People don't like to talk about.
We don't want to talk about no.
But it's this idea that there's another pandemic and there's this viral protein that we don't know, but it seems to be really important for virulence, and so.
Okay, I see. Okay, I don't want to keep that. I'll keep any other questions at the end if people have any questions.
Awesome. Okay. So now that we've talked a little bit about the research I've done over the last 7-8 years on epistemological beliefs, I want to segue into the Biology Everywhere project, which is really the meat of most of my talk today. So one of my first assignments as a faculty member was teaching non-majors biology. And every semester, I always know, regardless of what class I'm teaching, I always start with just asking students, what are you excited about this semester? What are you nervous about? Is there something about you I need to know? And over the course of many, many semesters, I had students telling me the same thing, that they were afraid to take my course, that they weren't any good at science, that they weren't smart enough to be there, that they didn't want to take it. And most memorably, I have learned to hate science. And that was the emphasis put in by the student. And so this was the foundation for a guest commentary that I recently wrote, and also my TED Talk. So all of these quotes up on the screen of my TED Talk were direct quotes from my students. And it wasn't just my students either. It was people I ran into in the general public. It was family members. And so universally, there's a lot of people that just don't feel good about their ability to do science. And this is hard for me to hear, both as a educator and also as a researcher, because it's like, oh, well, why do people feel this way? And where do these feelings come from? And what are the consequences of people just believing that science isn't interesting and that they're not good at it? So I went back to the classroom and I completely changed the way that I approached teaching general biology to people who were not science majors. I decided to make it more focused on daily life and how it intersects with other disciplines. And so why does a music major care about biology? And so we went from the beginning of the semester where students were saying things like, this isn't my strong suit, I'm not a fan of this, to the end of the semester where people were putting on their evaluations and their homework and sending me emails saying, this was my first positive experience in a science class. And so Biology Everywhere came about because I wanted to take that successful approach in the classroom and spread it to a larger audience. And so Biology Everywhere is 2 years old now. And then, as Maria mentioned in the introduction, I also have a spin-off children's book that's also built on these same foundations. And I'll talk more about both of those in a minute. So the Biology Everywhere philosophy is this idea that we present biology from the standpoint of daily experiences and as it intersects with other disciplines. So Biology Everywhere covers a lot of that same content you'd see in a general biology course, but the lens which we view it through is what's different. And so because it's told through shared daily experiences, it's more accessible and it's more relatable. And when it's more accessible and relatable, it helps people build confidence and change these narratives that they have about being good at science. So the more confident people are, the more engagement that they have. And that engagement is important for the health and welfare of society. So if you go back to my first slide with the epistemological belief section and talking about all these big science issues in society, we need people to be confident in their ability to engage so that they're more likely to engage rather than just listening to their favorite talking head, political person, and really get into these issues. Okay, so I want to give 2 examples from Biology Everywhere and how I do this. So the first will be an example from our daily experiences, and the second will be how biology intersects with other disciplines. So let's start with the decision to reuse. This is a big one that people are talking about on a very regular basis. This is one that my students kind of really latched onto as being very high interest. So real quick, in the chat window, what's something that you reuse rather than dispose of? So Maria is saying bags. Yes, I use reusable bags as well. And I've really tried to be cognizant about not taking bags when I go to the grocery store either. Gonna give it a few more seconds to see if anybody chimes in. Okay, well, if you think of something, feel free to drop it in, but bags is a big one. Water bottles, I use the aluminum water bottles. I really like those. There's lots of things that we can reuse. So I want to focus on bags a little bit because in the US, there's been a lot of press recently about people banning plastic bags. Some states already have. So when I was up in Alaska, everything was paper bags. I was visiting a few weeks ago. And so the question is, are reusable bags actually greener than plastic bags? Because the issue isn't quite as cut and dry as you'd think. So I want to pause for a minute and watch a short video. So this is a video I collaborated with an undergraduate student to produce. And it kind of gets into this issue of if plastic bags or reusable bags, which one is actually greener, and getting into some of the shades of gray around this.
Biology, or the science of life, is part of our daily experience. This video covers one topic that falls under ecology, a subfield of biology. Ecology studies how life is interconnected with one another. In fact, decisions you make on a daily basis can impact other organisms around you in many ways. Let's examine one of these issues a little bit more in depth: the decision to use a reusable bag when shopping.
You are at the grocery store standing in the checkout line, and a cashier asks you, you want to purchase a reusable bag today? The way I see it, there are 3 ways to respond. A, yes. B, no. Or C, have an existential crisis wondering if it's worth it. For the minority of you that chose option C, congratulations, you're in the right place. You are also in the right place if you answered yes and are now wondering what you're going to do with your 27th reusable bag that you will forget to bring to the store next time, just like you have the previous 26 times. Let's start by exploring our options. At any grocery store, we have our usual plastic bags. Maybe they also have paper bags. And lastly, we have our reusable bags. For the purposes of this, we're going to be talking about non-woven polypropylene bags, which is what most reusable bags are made out of. Let's assume that no matter what bag we use, we're just going to use it once. Obviously, that's not the purpose of a reusable bag, but just bear with me. When a bag is manufactured, it takes materials, energy, natural resources, and a variety of other things. However, once a bag is made, its environmental impact is not finished. At some point, a bag may end up in landfill or recycling or be repurposed. And what happens when a bag is no longer being used is still an important factor to consider. So we'll be looking at the life cycle of any bag holistically. Let's focus on 3 ways that it affects our environment. Global warming potential, which is a fancy way of saying CO2 produced, energy usage, and fossil fuel depletion. A single plastic reusable bag costs about 0.2 kilograms of CO2 to produce. Your paper bag is about 0.8 kilograms of CO2, and your reusable bag is about 2.6 kilograms. For all us Americans watching this, 1 kilogram is about 2.2 pounds, or about 9 quarter-pounders with cheese without the cheese bun or anything on it. The amount of energy it takes to make a plastic bag is 5 megajoules. Paper takes 23 megajoules, and a reusable bag takes 53 megajoules. For perspective, 53 megajoules is the amount of energy it takes to overcook your 20-pound Thanksgiving turkey by 30 minutes or so, making it just slightly too dry for your mother-in-law to approve. The amount of oil a plastic bag depletes in its lifetime is about 100 grams. A paper bag depletes about 200 grams, and a reusable bag about 1,100 grams. 1,100 grams is the same amount of oil that it takes to produce enough gasoline to travel 4 miles in a 2016 Ford Explorer or infinite miles in a Tesla, assuming you don't run out of battery. Now that you know this information, let's come back to the question at hand. How many times do you need to use a reusable bag to make it worth it? In comparison to a plastic bag, about 37 times. As a side note, I looked into cotton totes as well, and you need to use those 7,100 times before it becomes worth it.
What happens after you've used your bag of choice? Well, reusable bags can be, reused. In fact, I know of a reusable bag that has been in continuous use for over 10 years. Plastic or paper bags can be reused or repurposed as trash can liners, or if facilities exist, recycled. And paper bags can be composted or turned back into raw organic material. This is also known as dirt. However, the majority of plastic bags are never recycled. Waste Management estimates that only 1% of the 100 billion plastic bags that Americans use in any year are recycled. This means that the rest are taking up room in landfills or littering our communities, oceans, and closets. Plastic bags take 10 to 20 years to break down, and in that time, they leach microscopic plastics into the environment that can eventually work their way into our food and bodies. They are also suffocation hazards not only to us, but to other animals as well. The issue of which type of plastic bag to use at stores is one we face in today's society, and it's a pretty hot topic right now. Have you seen any of the headlines recently? There's a lot about plastic bag bans. Sometimes making green decisions isn't always straightforward. Whatever we choose to do, it influences the balance of life on Earth. Reusable bags take more energy to produce, but are more Earth-friendly in the long run. If you can remember to bring your bag with you to the store, that is. Plastic bags take little energy and money to produce, but shed microplastics, pose suffocation hazards, and tend not to be recycled, therefore end up as litter in our landfills. Sometimes it's hard to know what to do with this information, but the idea should be that whatever type of bag you have, to try and use it as many times as possible and repurpose it if you can. Do you even need a bag, or can you carry your items in your bag or purse that you already have with you? Or carry it to your car with your hands and then recycle.
If you found this interesting and would like more
So it's not as straightforward as we think it is. We like to think that, oh, you know, we have these plastic bag bans, it's going to be greener, but it's only actually greener if people can commit to actually reusing a product. And that applies to all reusable products. And there's also one other consideration to make with reusable products. And so usually I would do this as a Price is Right game. I decided to take the game out in the interest of time, and also just because I'm not sure what costs would be across international bounds too. But the idea is, how much does the reusable product cost versus its disposable versions? We have reusable cloth diapers, reusable sandwich bags, and also aluminum water bottles. And so what we see is that a single diaper costs about the same as a whole case of diapers. Same thing with the snack bags. Aluminum water bottles, single aluminum water bottle costs quite a bit more than a case of bottled water. And so what this brings up is that it's not just about resources used, it also becomes a social justice issue as well. Because we happened to cloth diaper my son when he was in diapers. And so you can get all the diapers that you'll ever need for every kid you'll ever have and beyond for about $500 US, whereas the cost of disposable diapers ranges to about $3,000 US per child. And so not everybody has $500 US just sitting around and can make it that big of an investment. And so what happens is that people get into buying the disposable versions and spending a lot more money in the long run. And so it's just interesting how many shades of gray that we see, and how this then fits in as part of an ecological system. And that gets into my last point, which is, oh, I can just recycle things. And so ecology is the study of interactions. And when we think about a bigger picture of ecology, it also interfaces with the economy and society as well. And so as it was mentioned in the video, a lot of plastic isn't recycled, and a lot of other goods aren't recycled because there isn't a market for it. And so that's why more companies are starting to come up with products that use raw recycled materials. And so Green Toys and Replay are both brands that buy previously used plastic and turn it into, in this case, toys and plateware. So they create a market for these reusable products, or rather they create a market for that raw material, because if there isn't a market for that raw material, even if people are taking it to be recycled, it's just going to end up in a landfill. Okay, so I took a slightly different approach with the Biology Everywhere philosophy when I wrote On the River. So this is a children's picture book. It's aimed for kids that are about 8 to 10 years old. It still has the pictures in it just because I feel like picture books have very broad appeal, but it also gets in kind of these harder scientific concepts as well. And so On the River is based on the river that runs through my town. So that's the Poudre River. You can see the Rocky Mountains in the background, and we follow the journey of 2 children. And I wanted these children to be someone who children reading the book could identify with, as well as that strong sense of place. That's why it's based in Colorado on a local river. And so we have Max here, and Max really likes model airplanes and really doesn't like science. He thinks it's hard. He just doesn't want to do it. And then he's got his big sister, Ellie, who's very excited about science, and she's earning her biology adventurer badge and wants Max to come along. And so here's her book with her checklist that she has to do, and then the badge that she's going to earn. And so they go out to the river, they find different organisms, they're trying to figure out if the river is healthy or not. And so one of the things they find is they do a macroinvertebrate assay. And, you know, this was chosen because it's a very high interest activity for children to do. The reason Ellie is holding a crawdad here is because I've sat and watched children catch crawdads in the river. So it's designed to be a way that students feel this strong sense of place, so they feel a sense of identity as a science person as well. And I also included a trail guide at the end so that students can go down and see these things for themselves. So things like osprey or fish hawks are very commonly seen here. See great blue herons all the time. Kingfisher, not so common. I've seen them before about 2 or 3 times, but I tried to include a variety. Okay, any questions on this part of my talk? Okay, so in the interest of time, I'm going to keep on going, but as always, if you think of anything, just drop it in the chat window. I've got other pauses for questions coming up, and there'll also be time at the end. Okay, so I had one more part on biology everywhere, and this is a part that I feel very strongly about as a musician, and that's how biology intersects with other disciplines. And so I want to talk about how art is foundational to biology. So just out of curiosity, do we have anybody here today who is an artist or a musician? So you can go ahead and drop in the chat window if you happen to do photography, if you sing. So I happen to be a flute player. I don't get to play quite as much as I used to, but I still get to play a bit. So do we have anyone else who's an artist or a musician here today? So Maria claims some of both. Wonderful. So I don't really identify much as an artist. I do identify as a musician, and I started tap dancing about a year ago, which is something I wanted to do my whole life, and I have a lot of fun with it now.
That sounds very nice.
Yeah, it's been a lot of fun. So I want to tell you a story about Santiago Ramon y Cajal. And he was a Spanish scientist, and he's also called the father of neuroscience. And he wanted to be an artist when he was growing up. And his father really pushed him to go into the sciences. And how he earned the title as the father of neuroscience has a lot to do with the fact that he made these wonderful drawings of neurons under a microscope. And so it's a great illustration of where science and art can meet together to lead to huge breakthroughs. So let's learn more. This is a TED Ed lesson that I collaborated on. I'm talking about the brain's biggest secret.
In the late 1860s, scientists believed they were on the verge of uncovering the brain's biggest secret. They already knew the brain controlled the body through electrical impulses. The question was, how did these signals travel through the body without changing or degrading? It seemed that perfectly transmitting these impulses would require them to travel uninterrupted along some kind of tissue. This idea, called reticular theory, imagined the nervous system as a massive web of tissue that physically connected every nerve cell in the body. Reticular theory captivated the field with its elegant simplicity, but soon a young artist would cut through this conjecture and sketch a bold new vision of how our brains work. 60 years before reticular theory was born, developments in microscope technology revealed cells to be the building blocks of organic tissue. This finding was revolutionary, but early microscopes struggled to provide additional details. The technology was especially challenging for researchers studying the brain. Soft nervous tissue was delicate and difficult to work with, and even when researchers were able to get it under the microscope, the tissue was so densely packed it was impossible to see much. To improve their view, scientists began experimenting with special staining techniques designed to provide clarity through contrast. The most effective came courtesy of Camillo Golgi in 1873. First, Golgi hardened the brain tissue with potassium bichromate to prevent cells from deforming during handling. Then, he doused the tissue in silver nitrate, which visibly accumulated in nerve cells. Known as the black reaction, Golgi's method finally allowed researchers to see the entire cell body of what would later be named the neuron. The stain even highlighted the fibrous branches that shot off from the cell in different directions. Images of these branches became hazy at the ends, making it difficult to determine exactly how they fit into the larger network. But Golgi concluded that these branches connected, forming a web of tissue comprising the entire nervous system. 14 years later, a young scientist and aspiring artist named Santiago Ramon y Cajal began to build on Golgi's work. While writing a book about microscopic imaging, he came across a picture of a cell treated with Golgi's stain. Cajal was in awe of its exquisite detail, both as a scientist and an artist. He soon set out to improve Golgi's stain even further and create more detailed references for his artwork. By staining the tissue twice in a specific time frame, Cajal found he could stain a greater number of neurons with better resolution. And what these new slides revealed would upend reticular theory. The branches reaching out from each nerve cell were not physically connected to any other tissue. So how were these individual cells transmitting electrical signals? By studying and sketching them countless times, Cajal developed a bold new hypothesis. Instead of electrical signals traveling uninterrupted across a network of fibers, he proposed that signals were somehow jumping from cell to cell in a linear chain of activation. The idea that electrical signals could travel this way was completely unheard of when Cajal proposed it in 1889. However, his massive collection of drawings supported his hypothesis from every angle. And in the mid-1900s, electron microscopy further supported this idea by revealing a membrane around each nerve cell, keeping it separate from its neighbors. This formed the basis of the NEURON doctrine, which proposed the brain's tissue was made up of many discrete cells instead of one connected tissue. The NEURON doctrine laid the foundation for modern neuroscience and allowed later researchers to discover that electrical impulses are constantly converted between chemical and electrical signals as they travel from neuron to neuron. Both Golgi and Cajal received the Nobel Prize for their separate but shared discoveries, and researchers still apply their theories and methods today. In this way, their legacies remain connected as discrete elements in a vast network of knowledge.
So I think the other thing that's really interesting about this story is that when, because they shared the Nobel Prize, and when they gave their acceptance speeches contradicted one another because Golgi was still set on reticular theory. And Santiago Ramon y Cajal was, of course, talking about the NEURON doctrine instead. And it's just another interesting expose in bias and how science really works, and that people don't always agree. And that's not the only example of the intersection of art and biology. So John Audubon set out to paint pictures of all the birds in North America. And so here's one of the rosette spoonbill, which is one of my favorites. I've seen these guys down in Florida before. And he also managed to document birds such as the Carolina parakeet, which have since gone extinct. Another example, if anybody's here who has a developmental biology background, you'll probably be familiar with either the Newcoop and Faber drawings of Xenopus. So that's frogs that are used as developmental organisms, or the Kimmel drawings of the zebrafish. So this is one of the original Newcoop and Faber drawings showing a frog embryo. A group in Boston at Tufts University commissioned a new set of drawings because they wanted more of these head-on descriptions, which were missing in the original drawings, because craniofacial abnormalities are a very common birth defect. And what's interesting is as the artist was working on this new set of drawings, they got new insights into how craniofacial structures develop. So it's really interesting that even today, in an age of quick and easy digital photography, we still see very important insights coming about because people can draw. And then also briefly, as we get into the last part of my talk on science communication, the arts are also a very important bridge to bring people back into the sciences. So we have people doing work with agar art. And so this is work done by a colleague down in Alabama, Sarah Adkins. There's also the work that Joel Sartori has done with the photo arc. There's the art people are putting together. I've seen several different art installations using trash to bring awareness to the plastic problem in the ocean. And then there's been music written as well. So I'm going to skip this question time and go on, since we only have about 10 minutes left, and I want to make sure we get through everything. And so I want to spend the last 10 minutes of the talk kind of as the culmination of all the work I've done over the last 10 years. So starting in graduate school, moving into doing the sci simulations and studying epistemological beliefs, to biology everywhere, to where I finally landed now with my work on how to engineer science communication. So I'm going to skip this activity for now, but I want to give it a second for you to think about it. So who is taking the evidence-based approach? Is it brain, who says you're wrong and I can prove it with facts? Or is it heart, who says you're wrong and I can prove it by being loud? And so the answer, depending on how you think about it, is either neither or both. And so facts don't change people's minds, and neither does talking loudly to someone. So if I started to shout at you, there's a good chance you'd shut down and not want to talk to me anymore. However, emotion can also be a very persuasive way of changing people's minds. It all depends on how you go about doing it, which is why you can say that neither brain nor heart is right and neither of them is wrong in this case. So about evidence. A lot of people get interested in science education and science communication because they get very frustrated by people who don't use evidence. And so oftentimes these are people from science backgrounds. So we think, oh, well, if they're a scientist, they must be really good at using evidence. Well, unfortunately, the scientists are human and have the same problems with evidence. And so case in point, this is the traditional view of education. We have the sage on the stage. We have the expert here. We have the crowd here that is listening attentively to this person talk. We now know that this is a very ineffective form of science communication or teaching, however you want to think about it. Science communication is pretty broadly defined. And so now we're starting to see more of what are called active learning classrooms, where we have small groups of people sitting at tables, talking together. And so instead of the sage on the stage, we have the guide on the side. Okay, but scientists don't do that. So this was a survey. This is about 10 years old. I've got a newer citation in just a minute. And if we look at STEM fields, so science fields, we find that most scientists still lecture rather than use active learning, and also do things like grading on a curve, which we also know is not evidence-based. And there was another study that came out in 2018 showing the same thing. So in this case, didactic is referring to this highly lecture-focused thing. And so we can see, for example, in chemistry and engineering, this is still what people are doing more than 50% of the time. They're not doing student-centered practices at all. Biology tends to be a little bit better, but that's still more than half the time. And it isn't limited to science classrooms either. So a paper I'm working on right now is on science outreach endeavors, very common science outreach endeavors that actually do more harm than good. And so this is with pinkified toys. So pinkified science toys for girls actually cause more harm than good, even though they sound kind of flashy and exciting. And also the most common science outreach thing I think I've ever seen, which is making things go boom. And so both of these things and actually promote disparities in STEM and opening up gender gaps, opening up gaps between students of socioeconomic status, and opening gaps between different minority groups as well. So they sound good, they're really not. And how we know that they don't actually look that good is by paying attention to the science literature. So what on earth do we do about this? Because it's really important that we, again, change these messages about science. That's, everything that I've done with biology everywhere. And so my big push right now, this is a grant that I'm working on, it's a paper that I'm working on, and I've given workshops on this as well, is about this idea of what if we engineered science communication, or SciComm. So this is where we get to the idea of learning engineering. So if we think of engineering in general, engineering is a field that leverages our understanding of how things work in order to create practical solutions. And at its core, that's what learning engineering is doing as well. It's an iterative process, and we take our evidence-based insights, our understanding of how we learn from the learning sciences, and then we use that to inform the design of educational experiences. So instead of using our understanding of physics and mechanics to engineer bridges, we take our understanding of how people learn and use that to inform the design of educational experiences. And so as an example, this was a bridge that formerly stood in Washington. There's a very famous video of when this bridge collapsed. It didn't last very long, and engineers found that they needed to allow for spaces in the bridge instead of having a solid deck. And so that's an example of where people were able to iterate, to be able to go back to evidence base and figure it out. And so it's the same idea of learning from previous attempts at things, pulling from the science of learning and using it to intentionally design experiences. So I want to give, with the last 5 minutes of my talk, an example of how we can do this when we design for bias. I've mentioned bias a couple of times throughout my talk. It's a very powerful force. It's a very interesting force. And a lot of bias comes in from how our brains process information. So estimates put this at about 950 billion pieces of information daily. And you can't pay attention to that much information. So only about 5% actually hits our consciousness, and everything else is handled through mental shortcuts. So I also want to point out that I just had a course on this come out with Lifeology. It's a free course that you can take if you're interested in this. All this artwork is coming from the artists that I collaborated with on this. And so if you've heard of Daniel Kahneman's work on System 1 and System 2 thinking, this is a way of explaining how our brain handles this. And so we have very fast thinking that is automatic. It's very emotional. And that's how we're managing on 95% of the information coming in, versus this System 2 slow thinking, where we can be a little bit more deliberate. And so I really like this analogy for System 1 and System 2 thinking that Jonathan Haidt came up with. And so you can think of your automatic thinking as being like an elephant, and your more deliberate thinking as being like the rider on the elephant. And so we would like to believe that we as the rider are in charge, that little bit of information that goes to our conscious brain is what's actually driving what we're doing. But sometimes the elephant takes over, and those automatic processes are driving what we're doing. And that's where a lot of bias comes in. And so biases or motives that we have can drive fast thinking. And there's a lot of places these biases can come in. They can be some kind of personal reason. So I live in a part of Colorado where there's a lot of oil field workers. And so they're very pro, let's drill for more oil. Because that's how they get their livelihood. It can be that in-group, out-group bias that I mentioned at the beginning of the talk with this idea of source. It can be about confirmation bias. This is what I already think. And so I'm going to continue to think this way. And so motivated reasoning is a type of biased thinking where we make decisions reflected on what we want, rather than scientific evidence. So if we go back to that comic, that's why presenting facts to people does not change their minds. It usually makes them dig in harder. And so just as an example of how we see this playing out is with mis and disinformation. So misinformation is when someone is unintentionally spreading bad information. They think it's true, but it's not. Disinformation is they're both concerning, but disinformation is a little bit more concerning because this is more like propaganda. This is the intentional spread of information that someone knows is wrong for some kind of nefarious purpose. So this could be trying to divide people. It could be politically motivated. It could be because you're trying to sell some kind of product. And in either way, we get false information being spread. And so the difference between the two is the intent behind that spread. And also the fact that we don't have penalties right now anywhere in the world for spreading disinformation. And so it's very easy to spread bad information now. How does this work? Why do people believe mis and disinformation? And it happens because, especially disinformation, it's designed to target people's motivated reasoning. It's designed to trigger motivated reasoning. It's designed to appeal to that elephant rather than the rider. And so that's why it can get a foothold. So what do we do about it? We need to get people to stop and think, because only they can change their minds. I get asked this question all the time. What do I do about this anti-science behavior and how do I handle it? You can't change someone else's mind. They are the only people that can change their mind. But what we can do is intentionally design activities that target the rider instead of the elephant, because we want people to slow down their thinking. We want to ask, Do things like motivational interviewing, asking open-ended questions, and then listening reflectively, repeating back, so that people are slowing down their thinking and less likely to trigger some of these biases and motivated reasoning. So part of the key with motivational interviewing is that people are more likely to listen if they first feel heard. Case studies can also be a really effective way of getting people to slow their thinking down so that they are reasoning through this stuff slowly rather than just having a knee-jerk reaction and digging in. Okay, so I want to thank all of you for coming out today and for your attention. If you'd like more information on my work, my science communication work, you can visit my website and subscribe to my newsletter. You're also more than welcome to follow me on social media. I'm always doing Biology Everywhere mysteries. And then I have 2 Coursera courses. So Biology Everywhere is a full specialization, and the science communication course is currently just a course. And one of my summer projects is turning it into a full specialization as well. If you're in the US, so you can get books on my website. If you're not in the US, I do have international distribution on Amazon. So I'm happy to answer any questions that you have. And again, thank you for being here today.
Thank you, Melanie, for this very educational talk. It was really interesting to see all these aspects. And my dad, was very happy to see Santiago Ramón-y-Cajal. As a neuroscientist, he's always my favorite.
It's such a cool story.
Yeah, actually, I was going to say it afterwards, but since I started with this, I think, in the old days, and I hope so now, but it's not so usual, I guess. If you are going to be an actor, you have to also have, a good voice. You have to cultivate all of your aspects of, your voice, how you talk, how you do everything. And as scientists, I think having the ability to draw, because old scientists had to draw all the time, especially anatomists, and taking good notes, because that was their only means of documenting things. So I think it puts you into a mode of paying more attention to what you see, because you have to. So I think you better understand what you're studying, and seeing the drawings of the hall is amazing, because we've seen this stuff under the microscope, and it's like literally it's the same thing. So I think that's something that we need to intensify these days, because we rely so much on technology that we don't utilize as much our memory part. That's a cognitive and neuroscience aspect, but at least that's how I interpret it. So that's one note. But actually, I have, so what would be your take-home message for, us that we sat through the talk, and or people that are watching your content? Like, what are the most important parts in biology education, like starting from early age, but even in older ages, because biology or education doesn't have an age, per se, an age gap in that.
Yeah, so that's a great question. And I would say the biggest take-home message from all of it is meeting people where they are. I think a lot of times as scientists, as educators, as communicators, there's this idea of you should do this or you should do that, and it doesn't work. And so it's paying attention to people's beliefs, it's paying attention to people's sense of identity, and it's paying attention to the literature on what works and what doesn't. And it all really fundamentally comes down to, I think, the answer to a lot of these scientific problems in society today is with meeting people where they are.
Okay, and with that respect, what do you do when the approach, even recommended by the literature that it works, does not work? I mean, how do you overcome such an obstacle?
So I think it comes down to some of the insights I was giving at the end of the talk and trying to understand why. And that was where I got into, this research program years ago anyway, too, is why do people feel this way? And what do we do about it? And I've talked to a lot of people and done a lot of listening, and there's just so many people, unfortunately, that I've run into that their experience with science is memorizing the periodic table. And so it's no wonder that they don't like it.
It's like the science periodic table. Yeah.
That's what they think it is that you memorize the periodic table and you dissect animals, and that's all biology, is just dissecting animals. And I'm like, no, there's a lot more going on than that. But that's the idea that people get. And then we have all these cultural stereotypes too. There's so many movies, so many books where the scientist is the bad guy.
Yeah.
And
Okay.
Yeah. And so then that plays into, oh, well, I don't like science. Science is a bunch of bad guys and people that are just trying to make money and control people. I mean, these are all common things I hear people say. Mm-hmm. And a lot of them are fear driven. And I think it starts with acknowledging that. I've got a copy of Merchants of Doubt sitting here. It's on my summer reading list. So if you haven't heard of it before, but it's talking about scientists who behaved less than well.
Below the bar.
Below the bar. So things like, going around and saying that cigarettes are good for your health.
Oh,
which we know now is not even slightly true. There's plenty of people alive who remember the Tuskegee experiments.
Oh, okay.
So people
so
have
it's real life. It's real life events. Yeah, I mean, unfortunately.
Yeah.
I think that's just the bad spectrum of human existence, I think. Either being a scientist or whatever. Tuskegee experiments are.
Yeah, so unfortunately, scientists have a bad reputation that they've kind of earned in many ways. And a lot of scientists, unfortunately, this is part of why I've pushed the science communication work so much, support these stereotypes and go out and try to do science communication endeavors that actually hurt a lot of people instead.
Yeah, because it's bad message and bad communication, like two sides of, I guess the approach. And also, have you because it sounds to me that you, in order to be able to transmit your message or approach your audience, you need a lot of psychology. I guess, knowledge. So do you interact or collaborate with psychologists, and what kind of psychologists would fit better in this situation?
Yeah, absolutely. So this is an interdisciplinary problem. That was the one slide I had at the beginning of my talk, because epistemological beliefs about science is what you would hear psychologists saying. Specifically, probably a cognitive psychologist or an educational psychologist. If you talk to someone who's from science education, they say nature of science.
Okay.
And there's some people argue quite a bit about if nature of science and epistemological beliefs about science are the same thing or not. I'm of the opinion that they're two sides of the same coin. I talk about that in one of my papers, that because nature of science kind of fits into more of the, what are the things that make science different from philosophy, for example, which is another domain of inquiry. Whereas epistemological beliefs are really getting at more of those cognitive structures of what do people believe, why do they believe that, and how do those beliefs act out in a classroom, for example.
Yeah, it actually goes into sociological issues, psychological. Yeah, it's interdisciplinary, as we just mentioned.
Yeah.
So another key question for all of us, I guess. So as a researcher, and you being an educational biology researcher, what will be your advice for people that want to give a presentation, a research presentation? What would be the key points that you would advise having in a presentation so people can get the message? I mean, I guess this will be breakdown like in a research audience, because that would be the higher end of knowledge versus a lay audience. How would you approach it in the two sides?
So I'm glad you asked this question, because it's something I'm really excited and passionate about. And then it's also something I'm going to have a hard time answering, because I do full day workshops on this exact topic.
Okay.
But I think what we know from the literature, and then just from my experience doing this and coaching a lot of people through this process, is that if you can write for the general public, you can do anything. It's a much harder skill set. And I've seen this in the literature too. People who can write well for the general public write excellent journal articles as well. And so in some ways, there's a very similar skill set. It comes down to a lot of difference in language and structure. So in a scientific paper, we have things like passive voice. We spend a lot of time building up to our conclusions. Whereas if you're writing for the general public, you use active voice and you get to the point right at the beginning, because that's what people want to know. I think both really come down to what's your take-home message, and less is more.
Okay.
You can make a much stronger statement without having a bunch of other stuff there. Those are very common comments I give to people when I'm coaching them through this process. And learning to tell stories. Yeah. That's a big one. We have evolved to like stories. That's why there's so many oral traditions, and people listen to their audiobooks and read books. And, there's no reason why we can't put that into oral presentation. So if you notice, my webinar today started off with the story of Alex and introduced this problem and conflict, and then how it's resolved through the course of my talk with my science communication work. People like stories.
So it came to mind now. Yeah. Sorry, go ahead.
Yeah, but I was just going to say, I've seen people do that in very high tier academic journals and tell these wonderful stories. And, I find myself citing those papers a lot more often because they're so engaging to read, as well as being really good science too.
You digest it more easily also, right? So I was going to say, it came to my mind that we have to become kind of influencers, because I guess the trend is you say a good story, you present it in a way that is digestible. So I think we have to get some lessons from that aspect of daily life now on. And apply it to our stuff. So thank you. It was wonderful. I don't know if you have any other comments. I don't want to keep you more.
No, I'm good. I really appreciate you having me. It's been good to catch up.
Yeah,
thank you everybody for being here today.
Thank you for accepting the invitation. It was wonderful having you and giving us all this information about how we teach and how we perceive it, biology. At the same time, and we hope to see you from close soon. I wish you a wonderful summer. Have a good rest after classes are done. Enjoy Colorado.
All right, well, have a good evening, everybody who's calling in. All right,
see you, Melanie. Bye-bye. Thank you everybody for coming. Bye-bye.